Good afternoon, everyone. I must commend the Honorable Chair for calling me up now because my ATP is even down due to severe hunger. All right, my name is Dr. Obinna Godfrey. My topic is unraveling the inhibitory potential of fatty acids from cola lepidota seed against leptin antagonism in obesity condition. Insights from quantum chemical analysis, pharmacokienetics, and molecular docking. All right, as you can see, this fruit is well known in Cross River State. So we use the seed for this investigation. What informed the use of this seed for this investigation is that it has not been extensively worked upon, like that of avocado pear seed. So for the research highlights, this study tries to address the pressing issue of leptin resistance in obesity condition, and the results we are promising. So these are I wish you can show that. These are the fatty acids we used in this investigation, and the leptin antagonist you are seeing there. The leptin antagonist you are seeing
Media,
there.
please, could you help us?
Yeah, very important. Please do that. The leptin antagonist you are seeing there is what is causing obese patients to eat, especially severely obese patients, to eat uncontrollably because they are not receiving satiety signal from their hypothalamus. So our interest here is to create an active site in the protein and dock it with our ligands to disorganize the amino acid sequence, that is, disorganize the three-dimensional structure of that protein and make it inactive so that leptin can bind to its receptors and the satiety can be initiated. This is another strategy to help obese patients because if we can handle this, that means that every other obesity management strategy can work. Exercise can work and all that because they feed uncontrollably, and the adipose tissue doesn't want to know because the more you put in calorie, the more it expands. It can never get saturated. So that's the concept behind this. All right. We used some computational measures to carry out this research, and you can see some of the software packages we use for the computational aspect of this work. Then the results, you can see the GCMS. I don't know why the young man is not projecting this. It's going to be very important if he does. The GCMS shows some prominent fatty acids that we use for this analysis. Then the Fourier transform infrared result showcased functional groups in the compounds in terms of drug design, in terms of drug investigation. To know your functional groups, the functional groups contained in the compounds is very vital because drug designers can manipulate the functional group. They are very important. Then the next thing we have here is the homo-lumo and chemical quantum descriptor analysis. The homo means highest occupied molecular orbital. The lumo means lowest unoccupied molecular orbital. Now, in drug design, you must have to know how your electrons transit from ground state to, excited state. The difference between the homo and the lumo gives you what is called the energy gap. Now, this energy gap gives you an idea whether your compound is reactive or not. If it is reactive, it means that it can be a drug. If it is stable, it means that within the system, it might not really interact with the living system. So a large energy gap, means that a compound is stable. A low energy gap means that the compound is reactive, which is okay. It can be a drug. All right. Now, For this diagram here shows the homolumo isosurfaces. What it means is that it is giving you an idea of where the electron clusters are, because in drug design you need to know where your electrons cluster, because when it comes to drug design you do a lot of manipulations. Okay, so here we also have a pictorial view of the electronic localization function, which gives you the respective electron clouds. You know, the more the electron clouds, I would have shown you this if he had projected it. The more the electron cloud, the more it is likely
Conclusions,
that,
please.
okay. Now, for the conclusion, I did pharmacokinetic, I did molecular docking. So let me just conclude. All right. Okay. In conclusion, the compounds were predicted to be absorbed via the intestinal cells, and they were well metabolized by the cytochrome P450 enzymes, and the linoleic acid and bis-2 ethylhexyphthalates were not HAG1 and HAG2 inhibitors. Then the molecular docking results showed that compounds interacted favorably with the receptor protein, and the bis-P and the LA had better binding affinity scores than the HSD and ODA. However, these binding affinity scores were low, with very few hydrogen bonds, thereby warranting more search for better natural products with greater scores against these leptin antagonists. Thank you very much for listening. God bless all of us.
Please clap for him.